In any case, this is an opportunity to kind of quickly go through the Center for Mesoscale Mapping, a P41 grant that we recently got renewed. I thought it'd be great to share what the grant's all about and let the leaders of the technology projects give a pitch as to what they're up to, hopefully to get people interested in collaborating with the new center. Okay, so a couple key questions. What is a P41? Some of you may not know that, what motivates our particular P41. And then if we have time, we can talk briefly about how the different elements all link together. So first, what is a P41? A biomedical technology research resource. So from the NIH website, it's just a grant funds centers that develop unique cutting edge technologies and methods to be shared with the broader biomedical community. And I think the real key is the sharing of these technologies with users. The P41 Center acts as a shared resource, making these specialized technologies accessible to the broadest possible community, training folks, and fostering collaborations. So that's from the definition, and I think we pretty much meet all the elements of that definition. So what are the key elements of a P41? It's kind of a complex beast. The key, the heart of a P41 are the so-called TRDs, or technology research and development elements. These are the core projects focused on creating novel cutting-edge tools, methods, or technologies, and you'll see those designed to meet emerging biomedical technology needs. The grant also has several other additional required elements. A very important one are the so-called collaborative projects. So the collaborative projects are really the test beds, which have the feature of both providing an opportunity for us to test these tools that are developed in the TRDs, but also to capture from the collaborative projects novel technologies developed by those sites and bring them into the P41. So there's this clear push and pull. Technology gets pushed from the TRDs, but also we pull from the collaborative projects. The service projects, another required element, are a little different in that they're really designed essentially for us to share mature technologies that have been developed from the TRDs to help external researchers solve their specific scientific problems. So a little bit further down on the hierarchy of importance, but still very important. And then finally, there's important elements in training and dissemination. And we'll also talk about that. How do we get the community to use our tools? How do we train them to do it? How do we share the results? If those of you who may be familiar with a program project grant, in some ways it's kind of like an inside out program project grant. The TRDs are like the PPG's cores, whereas the CPs and SPs are the kind of the science projects of a regular program project grant, or PO1. So kind of the same elements, but with reverse priorities. The scoring, the priority on a P41 is really focused on the TRDs, but they do need to be shared. So what about our P41, the Center for Mesoscale Mapping? What motivates it? Well, so why are we interested in mesoscale neuroscience? First, maybe a definition. This just comes from Wikipedia, where they were talking about mesoscale connectomes. So they say, rather than spatial resolution, or that corresponds to spatial resolution of hundreds of microns, and rather than connectomes like what Jeff Lichtman does, looking at individual neurons, or connectomes at larger scales, like we more typically do with our diffusion connectivity or a functional connectivity. This is really looking at distinct neuronal populations, either cortical columns or cortical layers that link hundreds or thousands of individual neurons. And that's the scale we're talking. So again, interesting, from Wikipedia, they said it still represents a very ambitious technical challenge. And according to them, can only be probed at a small scale, either with invasive techniques or at very high field MRI on a local scale. Our job here was to change this and basically tackle this technical challenge and be able to image at the mesoscale, ultimately across the whole brain. Modern neuroscience kind of looks at the brain in a couple different ways. A lot of neuroscience, if you go to the Society for Neuroscience, you'll see a lot of kind of bottom-up technology, individual studies of synapses, how individual neurons behave, how local circuits connect and use the individual neurons for distinctive functions. And of course, there's beautiful work. Our colleague Beth Hillman has shown these elegant microscale structures. Jeff Lichtman, of course, is building a connectome at the microscopic level, actually at the nanoscopic level, trying to look at essentially every synapse in the brain. So a lot of neuroscience is focused on this kind of bottom up. But of course, a lot of what we do at the center is really more kind of from the top down, if you will, looking at whole brain studies, maybe even collections of brains in social neuroscience, distributed networks. That's typically what we think of as the kind of functional connectivity that we do. Detailed system maps, like the very elegant maps that folks like Roger Tutel and Shaheen Nazar do, and the visual system. This is kind of top-down neuroscience. And of course, the top picture was from our original Human Connectome project, Connectome 1.0. The bottom slide is functional connectivity maps from our colleagues at the University of Minnesota. This is what I would call kind of the system level look, the top-down look at mesoscale, at macroscale science. But of course, the goal of neuroscience ultimately is to develop a comprehensive pan-scale understanding. And the fundamental question is, how and where will these two domains meet? If you go to meetings like the Society of Neuroscience, they don't tend to meet that much, right? The microscopic people talk to the microscopic people, and the cognitive scientists talk to other cognitive scientists. They're all in one giant room together, but less interface. And we think interesting things happen where they meet. And as a fundamental question, we'd like to be able to know kind of what are the emergent properties of these local and distributed groups of neurons that connect these scales at this mesoscale. That is the mesoscale where these properties begin to emerge. And the central question of the Center for Mesoscale Mapping is, how can we measure them? And of course, my feeling is if this comes from us rather than somebody else, we'll be in a position to define unique neuroscience at this scale of emerging local and distributed groups of neurons. And that's really fundamentally the mission of the CNM, is to figure out how we can measure at this scale. So let's just give a couple of interesting examples of what's happening at the mesoscale at the basic neuroscience level. Our colleague Anna Rowe has shown some super interesting work recently in macaques, looking at the mesoscale organization of dorsal and ventral pathways, the distinction even at the column level of distribution of columns for color, for luminance and overlays. And further, what she's been able to show is that there are distinctive connectivity patterns at a column level structure. In other words, these columns, distinctive functional columns, actually connect to distinctive columns in other parts of the brain. So the connectivity patterns that we see are not just reflections of kind of the map scale, but actually extend right down to the column level. And she's doing super interesting work with multi-channel infrared stimulation to show these connectivity patterns. But that's part of what we could be looking for. And of course, these are invasive measurements. We'd like to be able to measure these in humans non-invasively. Of course, there's the very beautiful and elegant work that Roger and Shaheen have been doing, looking at these kind of column level structures within the human primary visual cortex, showing excellent test reproducibility, and importantly, the individual variants at the mesoscale, which also suggests if we really want to understand this across subjects, we really need to be able to do it at the individual subject level, because the anatomical variance from person to person is quite significant at that scale. But this is also showing that, of course, these column level distinctive structures in luminance and 2D versus 3D and depth perception and contrast actually also sorry about that, also occur outside the primary visual cortex all the way up through dorsal and ventral visual streams. And more recent work from Shaheen has shown that this same kind of interdigitation that Anna was able to show in primates is equally present in humans. Beautiful studies showing that at the column level, stereo, motion, color are all represented within these map areas of, say, visual area V1, V2, V3, V4, but they are still distinctive anatomical areas when measured at the mesoscale. This is also now extending outside of the primary visual cortex to other parts of the brain. I show this. This is a very well referenced paper that talked about cortical hubs. This came from Randy Buckner and his group. I think, 1,400 citations. And, a few years ago, all the rage was the hubs, how there were these critical hubs that were active in a variety of different cognitive tasks, and they were key kind of coordination areas. But actually, if you talk to Randy today, he basically says, yeah, that actually wasn't right. Because what he went to do is to then go look at with very high sensitivity in individual subjects and show areas that were represented as hubs before are actually distinctive areas with distinctive activations across a wide variety of cognitive tasks. That's what these tasks are on the bottom right there. Interdigitated, like in the primary visual areas that we were talking about with visual stimuli, all kind of co-located and thus representing a hub at low spatial resolution, but actually very much not a single hub subserving the function of multiple different cognitive things, but distinctive areas for distinctive cognitive tasks. And basically, he very gently raised the possibility that there may be another alternative. If you talk to him privately, he'll say, yeah, basically, hubs, they're wrong. And here he's just showing that not only were these scales, of course, reproducible, but as we showed in the visual area, these hubs, while conceptually similar on an individual by individual basis, are quite distinct. And as a result, if you wanted to average, all you would see is kind of the blurry hub. You would miss this distinctive mesoscale function. So the challenge, of course, is not just to have the spatial resolution, but also to have the sensitivity to be able to do these studies in individual subjects. You can imagine that's technically very challenging. That's what we're about. Shaheen also has shown that not only is this of interest for basic neuroscience, but it's likely to be relevant in a very wide range of cognitive and other brain disorders. Here he showed, for example, in the visual system, looking at patients with amblyopia, that there's a remapping of the mesoscale selectivity in areas V2, V3, V4. And these are patients that are essentially stereo blind. And lo and behold, of course, the columnar structure has been remapped. And there are no longer these stereo specific areas. And again, showing in a wide variety of subjects that same pattern. Similarly, looking at the difference between strabismic and isometric amblyopia, that at the pattern of mesoscale activity is different. So this suggests that diseases that may seem phenotypically identical may be segregated based on their mesoscopic distribution of functions. And of course, that's relevant for prognosis and treatment. And we think that there may be many, many more examples of this. And just as one example is the really, I think, elegant work that Daphne Holt and Roger Tutel and their colleagues have done looking at personal space. These are studies that were kind of supported and motivated by what was happening in COVID and people's sensitivity around personal space. But basically, they showed in the parietal cortex, again, a distribution of columnar level segregated areas, but within a macroscopically homogeneous area, showing differences between approach and withdrawal, near versus far, and the distinctive mesoscale structures. And of course, their point was that this may also subserve disorders of personal space, which we see in a wide variety of psychiatric disorders. Now we may have the tools to be able to look for the mesoscopic disorganization of these functional areas. Now, of course, it's not just columnar level structures that we're interested in with Renzo Huber and his weekly layer group. It turns out the brain also has mesoscopic distinctive functions layer by layer within the cortex. This was an important study a few years back by Helen Barbers, who basically talked about structural motifs being predicted by laminar patterns of connectivity. So the different layers in different parts of the cortex connect in different ways. And basically, she also pointed out because these layers get laid down during the course of development, that the timing of an insult during development can specifically affect specific functional connections and thus the behavioral sequelae. And the point was that even with common genetics, if the insults are occurring at different times, you may get very different behavioral effects because the laminar connectivity patterns get laid down in a very stereotypic way, but distinctive way during the course of development. And she talked about one example, for example, looking at the differences behaviorally between schizophrenia and autism, which share many of the same genetic traits. It may very well be the differences in the timing and how these connectivity patterns get laid down layer by layer. Of course, if we can't measure those in humans, we have no ability to test these hypotheses. Now, hopefully, we can. And Kennedy et al. Talked about this, basically reinforcing the point that in cortical sample areas, there's a very principled matter which governs the rules of connectivity at the individual cortical layers, and that this could be very important for understanding both normal brain organization and a variety of diseases that we don't have a good understanding of today. Our colleague Earl Miller gave a talk at BrainMap last year, again showing the spectral laminar motifs of local field potentials across, in this case, the primate cortex. And again, focused in red, you can read that the laminar nature of the microcircuit will be crucial to establish. In his case, he wanted to use recordings as a common practice. He believed that the laminar understanding of connectivity and patterns of activity will be important for understanding their functions. We'd like to be able to measure those non-invasively in humans, something very important to do. And colleagues like Jyrki Aronen, along with others within our own program, have now been using laminar fMRI to study differences, for example, in top-down versus bottom-up influences in visual and auditory areas based on their unique laminar distributions. Brainstem, another area where high spatial resolution and high sensitivity is key. 700 nuclei, 300 subnuclei, a lot of work to do to segment it. All of you, I hope, are familiar with the beautiful work that Marta Biancardi has been doing, measuring both structural and functional connectivity of these, developing tools to make these measurements more robust, more reliable, extending the atlases to ever finer areas, to new parts of the brain, like the pons, an area she's working in literally as we speak. Again, a highlight of the need to move to mesoscale style measurements. And here's just, again, an example of behavioral disorder, REM sleep disorders, which again shows evidence of disruption when measured at the scale of these fine subnuclear structures. The final element that I won't have much time to talk about is we've also defined mesoscale as kind of a mesoscale in the time domain. Typical fMRI, of course, is time domains of several seconds. So, very kind of low spatial temporal frequencies. Electrophysiologic recordings, of course, can be much faster, but there is this mesoscale somewhere, between 0.1 hertz and say 2 to 5 hertz. And it turns out there's studies that are now showing at mesoscale temporal frequencies are unique frequency dependent functional connectivity patterns and functional cycling that seems to drive functional modules, the ability to press the temporal resolution of tools like functional imaging, and then combine that with activation tools to be able to really interrogate at a fine temporal scale is another feature of the CMM and another essential feature of mesoscale neuroscience. Okay, so, what are the kind of questions we might want to ask? What are the spatial scales of these emergent properties? What are they across the cortex? How do the structural and connectional motifs of these interdigitated networks actually look? How do they subserve function? Where do they exist and in what cognitive domains? Same is true for laminar structure and the functional differences that may be related to neurodevelopmental disease. Studying time-varying dynamics, especially at this kind of mesoscale, which, as folks like Laura Lewis have shown, is now feasible if we're careful and very sensitive in our measurements. And of course, exploring what happens in the brainstem and midbrain, where we have many of these tiny nuclei with extremely important connectivity and neurochemical properties that we'd like to understand. So just quickly pressing on, what are the key components? You're going to hear about these. Here are our four TR&Ds. You'll hear about these, so I'm not going to go through them. Here are the characters involved, really amongst the best technical group to work with. It's just a fabulous pleasure working with them. In a grant, of course, you always like to talk about connections between them. I'm not going to do those, but suffice it to say, lots of interesting connections between them, and you'll hear about those. The final thought: why do we do it? For me personally, it's a chance to sit around with amongst the smartest people in the center and really kick the tires on some new technology. I think for the center, it's an opportunity to really take these technology advances that individual groups are doing, think of them more holistically, and then apply them. And I'm hoping that what you get from today are ideas about collaborations that you'd like to develop with individual or multiple TRDs to advance your own neuroscience. And so with that, I will stop sharing. Clearly, I've gone way long, as I suspected I knew I would, but we'll let you guys carry on from here.
So we have a question, Bruce.
Hello.
Hello.
Yes, you can hear.
Hi.
Oh, there you are. Oh, I know you.
Yeah, I know you too. So I was wondering if you, and this is like, it can be speculation, if you have a disease or treatment of disease in mind where you think that, looking in the mesoscale and not the macroscale is really going to make, a big difference.
Yeah, it's an excellent question. I don't really have a great answer. I remember we talked about this a little bit with Jeff Lichtman during our retreat the other day. It was his intuition, and I hope he's right, that we're at whatever scale we look, we see interesting things and things of clinical relevance. We just haven't looked here before. I am interested, especially in the developmental diseases, these diseases that manifest later in life, but which clearly have roots that go back to early development. And these notions that, Barbas and others have talked about how connectivity patterns, at the laminar level might both be visible and may be reflective of, kind of the differences in the behavioral sequelae, because we know that the genetics is super overlapping in many of these disorders. So I think that's one domain. On the column level, I think we just don't know enough about what the column level organization of the higher order cognitive areas is, except that it is there. So, what do we look for? Smarter people than me are going to have to think about that.
Okay, so Bruce said it's like an inside-out PPG. I would say it's more like a set of linked R01s, but way more annoying. P41s come with huge numbers of requirements of SPs and CPs and SABs and site visits, and just see Susie will talk about training and dissemination. Except it's not nearly as much money as a set of linked R01s. It's like less than half the money you'd get for an R01 per project. And so why do we do it? We do it because in many ways it has formed the technical core of the center. And it forces us to work together in ways that, we would kind of like to, but we end up not having time for. Would Larry and I have ever kind of turned the scanner into a huge microscope? No, probably not without a P41. So we have three aims. The first aim is kind of improving the imaging at the microscopic scale with SFP and also with the Athinoula MRI. Aim 2 is to develop algorithms for vessel segmentation. And we originally got interested in vessel segmentation, not because we were interested in the vessels per se, but because we want to align the interior of broad homogeneous regions like the white matter with more accuracy. And so the vessels serve as a set of endogenous fiducial markers that we see everywhere in the brain. And if we can align the vessels, we have a better idea of, we have a better chance of aligning the stuff that's kind of between the vessels than otherwise. And then aim 3 is about building common coordinate systems. We're not going to talk about that today, but I think that's pretty cool, and maybe we can do that in a future brain map. So aim 1 is going to be Divya, who's going to present in a second, and then aim 2 will be Chiara, and that's it for me. So hopefully we can get closer to being on schedule.
Yeah. Okay. Hi, everyone. So for aim 1, okay. Yeah, so we're really interested in building a lot of quantitative OCT methods. Basically because, for example, we want to measure scattering coefficients because it's highly correlated with myelin content of the structures that we are imaging. However, we know that OCT data suffers from speckle. For us, it's noise. I know people use speckle as data itself, but we model it as like a multiplicative noise, which can be very tricky to remove. Furthermore, the OCT imaging system itself will have its own point spread function, a confocal point spread function, and a signal decay. All of these kind of affect the intensity and make it really challenging to estimate these parameters quantitatively. And last, if you want to do machine learning, we really don't have any open OCT data sets of the human brain. So we really tackled all of this in the solution I'm going to propose today by taking advantage of synthetic training data, and then really modeling both the speckle and the system behavior so that we can estimate not just the scattering coefficient, but also the imaging system parameters, so you don't have to measure them. And lastly, also remove speckle noise. The way we did this, yeah, so this is the schematic of how we did it. You can see here, we just synthesized like a segment, multiple segments. It's just an image with many tissue segments shown here. And then we assign the optical properties to it, run it through the optical model, get the synthesized images, then start putting in the speckle and other imaging properties to it, get noisy images, then pass it through a UNet, estimate the scattering coefficients, then pass it through an optical format model layer, estimate back the noiseless OCT images, pass it through a loss that really minimizes the likelihood, negative log likelihood of the speckle noise. So quickly running to the next. We first just implemented a 1D version of this network that I just showed you, just because we wanted to remove the spatial neighborhood information, just see if the network can optimize the model itself. And we found that actually did really well. These are 4 different phantoms, and the proposed method just completely was much better than the baseline approach that is generally used. And even with real data, actually, this is a hippocampus data set with absolutely no averages taken. The numerical optimization actually did not converge. It ran for days and it just never converged. It's that non-convex an optimization. The naive approximation, as you can see, yes, it gives us an estimate, but it's all corrupted with speckle. But our approach kind of did really well. It's fast. It doesn't require any additional measurements. There's no speckle there, and we haven't used any kind of spatial regularization here. So it's all 1D. So it kind of does a really good job of optimizing the model. And then we went further because we have the slab of OCT data, and we have one scattering coefficient. Instead, we kind of divide it into multiple because tissue changes in depth as well. So to just see how well it does in depth resolved scattering coefficient, and we found that it's way better, as you can see from the baseline. At much deeper regions where the signal is very low, we just suffer from, and the baseline method doesn't work. So this is very useful because what this means is you can cut your OCT slab much thicker, and that can be very useful in speeding up the acquisition. So we're kind of looking into that, and I pass on to Chiara to give you aim 2 updates.
Thank you. So we talked about the second aim, which is vessel segmentation. So Bruce already gave a bit of motivation why we are interested in doing it. In addition to vessels being interesting per se, we all know they drive the Bold effect, so they could be used to have a better interpretation of fMRI signal, etc. They can also be used to help registering images when they are at different scales. So here we see an example of 3 different modalities: ex vivo MRI, optical coherence tomography, and light shift microscopy. They have very different scales, so aligning uniform regions such as white matter can be challenging in those cases, but since vessels are everywhere, basically at every scale from 4 microns on, they can be used to anchor the registration and align in those regions better. So usually when we want to develop a segmentation model, we start by acquiring manual annotations. The problem is that for vessels, it's extremely hard. You see on the left a patch of ex vivo MRI. The resolution is 120 micron, and you can see that all those little moving dots are vessels, so it's very hard to get manual labels. On the right, you can see a OCT patch where we got some manual annotations, but no matter how accurate we try to be and how small the patch is, there will always be some vessels that are missed by the rater, and this was an expert rater. So how did we tackle this problem? We trained on synthetic data. So on the left, you see our synthetic vascular trees, and on the right, our examples of synthetic images with corresponding vessels. And as you see, we are not trying to be realistic, and that's on purpose so that our model is exposed to as much variability of data as possible. And the advantage of doing that is that now our training data are perfectly paired by construction, so we don't have the problem of missing vessels in our manual annotations that we would have when using real data. So we use the synthetic data, we train a model, and for the sake of time, I will just show qualitative results, but I promise we validated this. So we developed vessel segmentation for different modalities. So here we have an example of an ex vivo MRI hemisphere, and I'm showing all the vessels contained in a 15 millimeter slab centered around the display slice, and they are color-coded by distance. This is a way of sort of visualizing some vessels in 3D without being too overwhelming. So we see some nice patterns of diving vessels. Another modalities that we were able to segment vessels on is time of flight. So this is in vivo angiography. We typically see bright arteries based on the inflow effect, and we got working segmentation on these ones too. This is an OCT patch. Here vessels look quite different. They are darker and bigger because it's a very much higher resolution. We see some results here. And lastly, I'm going to show this nice 3D API, thanks to Renzo, where we see some dark veins. And even though our method was never originally developed or fine-tuned or specialized for this type of modality, it works quite well. So we can see that it's very flexible. It's going to be on GitHub very soon. So if you're interested in segmenting some vessels, check it out or reach out to me. And concluding, we saw that realistic modeling doesn't seem to matter. And this is in contrast with the first aim where Evia talked about, where we need to have a more realistic model because we need to actually estimate some tissue parameters or like the scattering coefficients. And since the network has never seen real data, it generalizes quite well.
All right, so for TRD2, we have Risha and Birkin presenting on acquisition technology for in vivo functional and quantitative MRI at the mesoscopic scale.
Right. TRD2 is about acquisition technologies for in vivo functional and quantitative MRI at the mesoscopic scale. And there's two aims in this TRD. One, the aim one will be focusing on mesoscale fMRI with high spatial temporal resolution and fidelity. And aim two will be focusing on quantitative MRI at the mesoscopic scale. And I'll be talking about the functional MRI part first. Then the Birkin will be presenting the quantitative MRI. So with this TRD, we'd like to develop mesoscale fMRI with high spatial temporal resolution and fidelity. And with the last cycle, we've successfully developed novel acquisition technologies that provide a distortion-free multi-echo imaging, that provide standard fMRI or layer fMRI with improved functional sensitivity and specificity, and that have been successfully disseminated to research institutions globally. And in this cycle, we will be developing technologies that achieve mesoscale fMRI with significant leap in spatial temporal resolutions and coverage beyond the current limits. But we will also be addressing key challenges that impede spatial temporal fidelity and robustness. We will be developing core technologies, including this 3D tidy technologies that involve synergistic design of novel acquisition and reconstruction technologies to enable high acceleration and SNR. We'll be, kind of leveraging efficient readout and high dimensional encoding. We will develop methods to address motion and bezier robustness. We will also develop, contrast with microvector specificity. And with the successful development of this technology, our goal is to achieve ultra-high spatial and temporal resolution while achieving high anatomical integrity, improvement in TSNR and RCNR, and resolve mesoscale functional features. This TRD will have a lot of interactions with other TRDs and collaborative or service projects. So here are some examples of what we can achieve with this TRD. First is the mesoscale fMRI with high spatial temporal resolution. Here show some example where we can achieve 500 micron isotropic fMRI data with whole brain coverage with volume TR of only 0.9 second. Or here shows the data with 340 ISO resolution with a very short volume TR as well. You can see the high data quality that reveal these detailed structures across these different slices. And at this resolution, we can begin to see these highly detailed structures, including shown here is live genera in the visual cortex with very thin myelinated layers. And it's very important to achieve mesoscopic resolution because, as you can see, these structures can only be seen when you go down to mesoscopic resolution below 500 micron or even to 300 micron. And at the standard resolution or even this sub-millimeter resolution, these structures, are blurred out. And we also aim to kind of achieve high quality to begin to see these more challenging areas better, including the hippocampus and also the temporal lobe better with the development of new technologies. In addition to spatial resolution, at the same time, we'd like to achieve high temporal resolution as well. This is a spectrum show for the 500 micron data where we achieve 0.9 second TR. And you can see that even at this high spatial resolution, we can achieve high temporal resolution that nicely resolve the respiration signal here. And we can go to another kind of protocol or spectrum where at standard resolution, whole brain coverage, we can achieve really fast TR of 150 millisecond. And at this TR, you can see that we can not only achieve, resolve low frequency brain oscillation, but also respiration and cardiac and their high frequency harmonics. And this will enable us to see faster brain dynamics, but also be able to, remove these noise confounding noise factors and achieve clean, better functional connectivity. In addition to spatial temporal resolution, we'd like to achieve kind of high data fidelity, including achieving kind of a distortion-free imaging to achieve similar kind of anatomical integrity with MP-RAGE or structure images. And also the benefit comes also with eliminating the dynamic changes due to the field changes across the time series as well. And it's important to also at this resolution to preserve functional sensitivity as well. With the development of new acquisition and reconstruction technology, we'd like to preserve functional kind of sensitivity at this extreme high resolution as well. And we'd like to kind of validate it across multiple subjects. And not only for task fMRI, this high sensitivity will enable us to see resting state fMRI within a short scan time, but be able to delineate better kind of connectivity with better spatial specificity as well. And finally, not only to look at the structural kind of detail structure, but also, the important is to look at the functional connectivity, detailed mesoscale features, including the functional activity across different layers that has been demonstrated to be better when you improve the spatial resolution. As well as looking at these more advanced kind of columnar or advanced mesoscale features, including the motion selectivity stripes that are analyzed by Shaheen. Okay, to summarize with this aim, we'd like to develop mesoscale fMRI with high spatial temporal resolution, with high anatomical integrity, preserve functional sensitivity, and be able to finally begin to see these advanced mesoscopic features with better fidelity and capabilities. With that, I'll pass it on to Birkin to talk about the quantitative amount.
Great, thanks, Fushu. So in the previous cycle of P41, we had contrast-weighted imaging that was qualitative. So this new focus on quantitative MR is new to CMM. What we want to do is to provide specific information on tissue composition, which is really only available through in vivo quantitative MR non-invasively. To be able to do this, we want to create new technologies so that we can overcome physiologic and physical barriers to be able to provide this efficiently at high fidelity at the mesoscales. In the first sub-aim, what we want to do is quantitative relaxometry, T1, T2 mapping using an open source PulseSeq sequence programming environment, and also incorporate volumetric navigators in the unused sequence time so that we can gain robustness to subject motion. Our initial goal was 450 micron, but as I'll already show you, we surpassed this resolution target for ourselves. So the sequence itself is super simple. We have a T2 preparation and an inversion pulse to give us sensitivity to T1 and T2 variations. And then we just do a flash readout, which is the same thing in an MP-RAGE. So why do we want to do this specifically? Because there are already multi-vendor implementations that is good for dissemination across different sites. We also have good momentum in this dimension. Shohei won the Young Investigator last year with his open source harmonization paper. Fingerprinting is also a really nice way, but at 7 Tesla, B0 related blurring as well as trajectory correction might become a bit more challenging. So this could be a powerful way to go to mesoscale resolutions. Couple things we need to address is the T2 preparation needs to be adiabatic so that we can be resilient to B1 plus artifacts. And the same thing is true for the immersion pulse. The next thing that we develop with Rob and Borjan is to incorporate these volumetric navigators at the end of the TR. These are very low resolution, 8mm isotropic, but they give us the ability to detect a sub-millimeter level subject motion within each TR. So taking the sequence from 3 Tesla will not directly work at 70 without significant modifications. As we described, we need these adiabatic signal preparation pulses, which are quite SAR intensive. We also need to do self-supervised AI reconstruction. This gives us increased SNR and image fidelity, as I'll show you. And as Divya also explained, we can also use self-supervised AI for parameter estimation to be able to invert non-convex and complicated physics models so that we can estimate the parameter maps out of the acquired encoded data. And finally, as I talked about, we need prospective motion correction so that we can address these physiological confounds. In terms of the second step, we use this joint zero-shot self-supervised learning reconstruction. So what this means is that there is no training data set, just like this fast MRI data that you might have heard about. In this case, reconstruction and training is done entirely using single subjects data. And in this case, we also train a single model that generalizes across the entire volume so that we just train a single model that works across the entire whole brain data. So with this, we can already improve the image fidelity quite a bit. So in the upper row, you can see these contrast-weighted images from which we estimate the parameter maps. So with compressive sensing, they look quite reasonable, but with this self-supervised AI reconstruction, we can further improve the image fidelity and SNR level. In terms of parameter estimation, after we get these contrast-weighted images, we need to invert our block equations and estimate the underlying parameter maps. This is exactly the same thing that Divya explained, so there is good synergy between TRDs. So what we do is just put a CNN in between the acquired data and the parameter maps, and we directly hallucinate these maps. And then we go back and synthesize these contrast-weighted images by incorporating the block equation, so we can fully incorporate the physics into these parameter maps. There are some additional details, such as inversion efficiency. So during the lengthy inversion pulse, there is some signal relaxation, So if you don't take that into account, your parameter maps might be biased. So we can also estimate this jointly, and we also put a B1 plus map. These are some details, but they are important to boost the accuracy of the maps that we get. So putting everything together, we can finally get 390 micron isotropic parameter maps in around 44 minutes with prospective motion correction. This is in Bay 2, and if you're interested, please let me know. Next steps, we want to characterize the repeatability as well as the accuracy, which is challenging at 70, but we'll work on that. And there's some B1 plus issues that are remaining, especially in the back of the head, in the cerebellum, that you might be able to see, and we will try to tackle those as well. Next, we want to also look at QSM and T2 star mapping. For this, we want to do a multi-echo gradient echo type sequence and aim for 450 micron isotropic with motion correction. So what do we need for this? We need high acceleration so that whole brain coverage is feasible, but without sacrificing G factor SNR loss. We need to correct for motion and dynamic field changes without slowing down the acquisition so that we can boost the fidelity. And finally, we need to make sure that there is enough SNR after doing all this stuff at mesoscale resolutions. So what we did so far is to incorporate these wave encoded acquisitions into a pulse sequence. This is at 3 Tesla and at low resolution, 1 millimeter isotropic. You can see with Cartesian encoding, you get quite a bit of image artifacts, but with this wave encoding, we can nicely clean it up at high accelerations. In this case, we have multiple echoes, and we can jointly reconstruct these as well by subsampling the case space differently. Next steps is to incorporate flow compensation for vessel contrast. And we also began to deploy some of these AI reconstruction techniques. So putting these together with Rob, we were able to do a 350 micron isotropic QSM acquisition at 70. Here, each acquisition is 15 minutes, and we did 3 different head orientations so that we can invert the QSM's forward model more properly. There is motion correction, but no dynamic B0 updates yet. And you can begin to see quite nice details, such as perhaps these increased myelin in layer 4, as well as these mammolothalamic tract, which Susie actually identified for me. This is all nice. What are the next steps? You want to go to wave encoding from Cartesian. We already have volumetric navigators, but no field updates. And we began to see the benefit of this AI reconstruction. You can see in the bottom row, it helps boost the SNR, but all these QSMs were coming from standard vanilla parallel imaging reconstruction. And we finally got some 250 micron isotropic. I managed to get these like 2 hours ago, but I wanted to show anyway. So this is whole brain coverage. I'm just showing one slice. And this is standard parallel imaging reconstruction, nothing interesting. So this took about 40 minutes, again, using prospective navigation. Renzo told me that this might be the highest resolution in vivo gradient echo yet, but that needs to be verified. But I think what's interesting is there is SNR to burn. I think we can go below 200 micron, and I think that's our goal after this. But why do we want to do all this stuff? We can make use of these T2 and T2 star maps to be able to derive quite specific contrast using the source separation QSM idea. So this allows us to disentangle cortical myelin and iron from each other. And I think that is really the only way you can do that non-invasively. But it unfortunately requires an R2 prime map. So both T2 star and T2 are needed. So far, we don't have that yet. But what we did was using this R2 prime net method to estimate an R2 prime map from the acquired R2 star data. And we can begin to disentangle these diamagnetic and paramagnetic contributions to QSM. So current maps are a step towards the desired contrast. And hopefully, as we begin to get the T2 maps, we will finally incorporate the correct physical model and be able to disentangle these contributions. I think that's it from us.
So in the interest of time, we're going to move on to TRD3. This is basically a new aim for the Center for Mesoscale Mapping in this cycle, tools and technologies for high fidelity mesoscale diffusion MRI. And so I will be presenting this alongside Larry for the high fidelity diffusion imaging. So the first aim of this TRD is really focused on developing efficient acquisitions for high fidelity, high resolution diffusion MRI with really rich information. I'm particularly excited to present these results by Zijing and Felicia on Romer EPTI. This is a new version of the EPTI method that Felicia presented initially with our first cycle. It was really fleshed out in both TRDs 2 and 3 for this cycle. So with this acquisition involves a rotating view motion estimation kind of approach that gives us very high signal to noise ratio and also enables, as a result, high spatial resolution acquisition. So you can see that really beautifully on 2 of the systems here, both at 3T Bay 4 as well as our Bay 2 70 Tera, now Tera X. So just a few illustrations. I'll go through this pretty quickly to see if we can catch up on some time, but you can generally see how well this motion aware recon does in the brain, really allowing you to see structures that you wouldn't be able to see in somebody who is moving. And I'll also mention as an aside that these methods can also now be deployed to other body parts outside the brain, including the body. So more to come on that, I think, in the coming years. With the EPTI readout, we can also get multi-echo time information to facilitate diffusion relaxometry measurements. And I think really leveraging the complementary information provided by relaxation contrasts as well as diffusion will allow us to kind of get new information from the tissues. Zijing and Felicia have incorporated a deep learning accelerated framework into the Romer EPTI acquisition to really accelerate the acquisition. And in the next couple of slides, I'll show some of those benefits for the resulting acquisitions, so at 3T and 7T. So here you can see the 500 micron kind of 4 times accelerated acquisition that can be done within 20 minutes on the Prisma while maintaining high fidelity and high information content. And then on the Connectome 2 system in Bay 8, they've been able to push the sequence down to the sub 400 micron level. So really matching what we can do in ex vivo tissue and beyond. And as you can see here, I think taking a cue from Birkin, there's SNR to burn. So very excited to see what we can do here, not just for this type of acquisition, also for multiple B values, especially high B values. And so as you can see, there's various structures you can identify, including all those fine fibers going into the gyral crowns within the U fibers using this method. And then finally trying to see if we can incorporate other strategies in the within this cycle of the TRD2 or in the TRD3 to address stronger field imperfections, which we anticipate will be the case, especially with these higher performance gradient systems, including the Connectome 2 and then the impulse scanner, which is coming later next year. Working together with other investigators within this TRD, Felicia Zijing they're collaborating with my group to really see if we can target efficient microstructure imaging using diffusion relaxometry, as well as biophysical modeling to extract information such as kurtosis within the gray matter. We're now looking at kind of more biophysical models like estimation of cell size, cell density, axonal diameter, and density. So with that, I'll turn this over to Larry and Jason to discuss the AIM2 goals.
Great, thank you. So that was a little bit about bringing diffusion to 7T. This whole TRD is about that. And in AIM2, we're really focused on the more traditional 2D slice selective, maybe single shot diffusion. And the real barrier to making that work well at 7T is the B1 plus and B0 inhomogeneities present at 7T that are just enough worse than at 3T to make it a real problem. So our aim is to create the excitations that are needed to overcome these problems. And Jason is working on the shim array needed for the B0, which also feeds in, as you'll see, to the B1 plus problem. So for the excitations, we've kind of converged on these tailored excitation pulses. And in a tailored pulse, you pre-compute a so-called universal pulse on a data set or a whole bunch of individuals. This is pre-computed. So it's sort of a one-size-fits-all pulse that best handles all these individuals. But then the patient enters the scanners, you acquire their B0 and B1 field maps, and then you essentially tweak the universal pulse to tailor it to the individual pulse. So the UP is the starting point of the optimization. And one of the directions that we're very interested in is using arbitrary trajectories for these pulses to give more degrees of freedom to the optimization process rather than just vanilla parallel imaging, parallel transmit. And you can see what this does to the excitation K-space during the excitation. So that's what is happening during the RF pulse. And you have to optimize this trajectory as well as the pulse, subject to all the standard hardware constraints. And it's a very difficult optimization problem. And there's a lot of variables because you can do any trajectory you want, obviously. That's a lot. And so this makes it all very hard. And one of the things we've been looking at is rather than optimizing each time point in the waveform as an independent variable, which is a lot of time points, is trying to compress that using polynomial basis sets and then compressing and then just optimizing the parameters of the polynomials. And this leads to all sorts of other problems in how you calculate the pulse updates. And John Drago has been working on this and has made good headway in the optimization framework to do this, and has starting to show some good slice selective optimizations at 7T, both in the test population and then of the tailored pulses themselves. And here's an example, and the advantage of compressing is it just goes so much, the optimization goes faster than the piecewise constant basis and gets a slightly better answer. The other approach we're taking is a so-called two-photon excitation approach, and this sort of invites the B0 shim array into the pulse excitation party. The goal is the same, to try to flatten out the flip angles of, say, a slice selective 90-180 pair. And but now we want to take advantage of the shim arrays. And the way we do that is by switching from the conventional excitation depicted here, where the RF connects to two different up-down state energy levels of the spin exactly on resonance, so exactly the right frequency. And then the spins in the rotating frame just go straight down to the XY plane. In the two-photon case, you excite with your RF bird cage, but slightly off resonance, maybe 10 or 20 kilohertz off resonance. Nothing happens. That's a little simulation of the block sim of what happens. It stays along the z-axis. But if you provide the additional missing energy with another RF oscillating field at the missing, say, 10 kilohertz, then in fact you do get an oscillation. It's maybe about 60% as efficient at best. But nonetheless, it's now capable of doing it. And that second oscillating field is provided by the shim arrays. And the beautiful thing about that is that we have 32 or 48 shim channels. That's far more than the parallel transmit channels. Normally, parallel transmit channels would cost $100,000 each. The shim array amplifiers are something on the order of $100 each. The other thing is it doesn't add any SAR since it's low frequency. So we're doing the same thing. And here John showed that these tailored multi-photon PTX pulses can achieve a really good parallel transmit excitation. And then Jason, of course, they use the shim array. And if we want to marry these two approaches with the arbitrary waveforms on the shim arrays, we need to really dynamically update the shim arrays. And this is what Jason has been working on to allow these shim amplifiers to essentially slew to the desired waveforms and with a high fidelity, decent noise performance, etc. So he's worked now on multiple revisions of the amplifier to give them this capability. And finally, this is just a fun test he did where he took a 32-channel shim array and formed a mode that looked like a linear gradient and made an image with it. Just instead of the normal MR gradients, he used the shim array instead, which is now fully switchable. Not a super high resolution image because they're weak gradients, but that's the approach for AIM 2. Thank you.
Yeah, no, thanks so much, Larry. And AIM 3 of this AIM basically rounds out all of the data acquisition and basically pulse design efforts with tools for interpreting mesoscale diffusion MRI with advanced simulation and parameter estimation tools to actually interpret what the information content in those diffusion MRI data reflect at the tissue microstructural level. So with that, we'll turn it over to TRD4.
So yeah, I'm assuming people still are sticking around, so I'm just going to go quickly. And our TRD4 is about linking biophysical modeling with multimodal brain stimulation and recording technologies. So just to give a background, we really nowadays think that most of the neuropsychiatric illnesses are disorders of brain networks, and TMS can be used and has been used and has been approved by FDA for multiple indications, but there's really not a clear picture yet why some people respond, what to do if they don't respond, and what is the optimal protocol and dose for personalized treatment. And here are some of the technological challenges that we've identified, trying to really understand what happens locally at the stimulation site, use multi-scale modeling, and then try to modulate multiple nodes of the connected networks with multi-channel TMS systems. And finally, we try to measure the TMS elicited responses with high spatial temporal resolution with multimodal imaging using TMS, EEG, and fMRI. Here's our team. We have a very broad expertise ranging from RF coils to fMRI and EEG and TMS physics. And here are proposed new technologies. We have a computational modeling aim where we try to go from the macro to the meso to the micro scale in terms of trying to model the electrical fields. We're also developing new types of multi-channel TMS systems so we can try to verify these models. And finally, we're going to do multimodal imaging using multi-channel TMS combined with EEG and fMRI. So just to give a couple of, bites of what we're doing right now. For AIM-1, we've done a sort of verification of the E-field models because it's really important for our project to have a very good control of like how strong the stimulation is in the brain. And we've done some work on the microscopic formalism together with Sergei Makarov. So just to give like a quick idea, the models themselves are actually quite complex. There's a lot of data going as an input, like the models with the coil in the head, the numerical solver, and so on. So we did a little bit of a verification. So we did the motor mapping with 2 different coils using neuronavigation. And with the 2 coils, we try to predict one from the other by repeatedly measuring the motor thresholds. And here's the results. So it is actually, if you just look at the motor threshold as a function of the stimulator output, there's quite a bit of difference because the small coil requires higher currents. But then if we calculate the electric fields and compare them at the motor cortex, this really gets equalized out and the correspondence is very high. So what we can do with that, we can establish a good relative measure, so we can use this to test the predictions of the models. So we can take the models from electrical stimulation or magnetic stimulation, then try to couple these with the realistic neuronal ensembles, and we can kind of use the electric fields to offset, let's say, from the electrical stimulation. We can modulate the threshold and see how the neural models would predict the thresholds to go. And with this way, we hope to gain insights of how these methods actually work. Now, going to AIM-2, the goal here is to develop a more advanced multi-channel TMS system for more flexible control of the stimulus and to be able to do repetitive TMS experiments. So for this, we recently did a physiological verification of our electric field steering with multi-channel TMS. So this was done by Mohammed, and we used 2 3-axis coils where we created 2 different shifts and a centered electric field, so we could test whether shifting this pattern actually results in the corresponding shift in the physiological data. We had 7 scalp targets, and we measured all of these 3 coils shifted pattern, virtual coils, which we call them, to get 21 estimates of the resting motor threshold. So here we can see that when the shift was 0, we get the symmetric pattern, and then when we apply the shift and drew the motor threshold, this is the 1 over the motor threshold. So there's a peak where the motor threshold is the lowest. And we get these nice shifted patterns, and they kind of shift with the expected amount of 1 centimeter. And we did this for 3 subjects, and when we average them, it's quite nice correspondence with our electronic shifts and our physical shifts. The paper is currently being submitted. And finally, a couple of words about combining the multi-channel TMS with EEG and fMRI. This is Lucia's work, was recently published in Imaging Neuroscience. So Lucia's project is to developing this so-called flexible RF cap, which can be then used to put the TMS coil anywhere, literally. So it gives a lot of degree of freedom to position the TMS coil. And here we did our first in vivo experiments where we show an example of single pulses of 110% of resting water threshold and compare that to voluntary finger tapping task. And indeed, we get very nice quality data and highly similar activation patterns as we expect. And of course, Lucia is working on this as we speak, but the next step is to try to integrate the EEG with the RF cap to enable this triple combination. And this is, of course, something that it then can be also used for closed loop applications. So thank you. That was my part.
New aspects of training and dissemination for this second renewal cycle of the P41. So in the training aspects, we basically are partnering with the workshops and short courses offered through the Martinos Center to add a bit of a mesoscale flavor. And in some cases, the new courses that we're offering here or continuations thereof have much more of a mesoscale focus. So I think one very nice example of this and highlighting the laminar fMRI analyses that have been brought to our center, thanks to Renzo Huber, who's joined the center, the Martinos Center as of June, and is now also integrated into the Center for Mesoscale Mapping. So Renzo held the laminar fMRI course as part of the Minnesota workshop, a workshop from another P41 to demonstrate synergies between different P41 centers. And we had many faculty present who are involved in P41, including Renzo, Anna, and then also John, kind of a P41 alum from MGH. And there are additional kind of center-wide trainings on advanced acquisitions that Renzo and the whole ultra high field team have brought in since the summertime. So including kind of this PTX workshop with Sydney Williams in Nottingham, advanced B0 shimming on the scanner with our Health and Years collaborators, and then also a PTX at 7T with Patrick Liebig, also from Siemens. So yeah, many thanks, especially to Renzo for providing these slides and also just kickstarting all of these initiatives to help us train a broad set of users. We're also continuing to be involved in the annual connectivity course. Unfortunately, this year we could not hold this course, but we will resume, I think, in 2026, and we're actively looking to collaborate with people on reviving the course with a mesoscale emphasis, highlighting how users can make use of all the really awesome technology we've presented here today. The TMS Core Lab recently completed their training in November. Here are some just of the highlights that this annual training course provides. And it's basically a way for us to teach people how to do TMS, but also integrate the advanced TMS technologies that are being developed by TRD4. In terms of dissemination of our technologies, our software and hardware dissemination is being kind of actively pursued through multiple channels, including sharing of code, pull sequences, and tools on websites, and also kind of integrating directly into Siemens C2P packages, as well as ultimately, hopefully, in the hardware they're developing for the next generation systems. As two examples of recent technologies integrated into the Siemens product EPI sequence, this dual polarity grappa developed by John Palomani. This has actually made it into the Siemens, the latest software version, their sequence for fMRI, as well as for diffusion MRI, as well as the Fleet ACS feature that he developed as well. We are currently planning for at least 2 symposia, I think, during the cycle of the P41. So building on the success of the Mesoscale Mapping Symposium that we held in 2023 in collaboration with the Neuroimaging Training Program. That seminar was focused on bridging scales and modalities in neuroimaging. Would love to involve all of you in this symposium as we start to brainstorm and plan for the next iteration. And finally, we have the website, the CMM website, which you can go to find out the latest developments and also learn more about becoming a collaborative or service project with our center. And I think I will leave it at that, I think, just to, in the interest of time. So thank you all for joining and for staying late, for those of you who were able to stay. Happy to field any questions. This will also be posted on the website for further future reference. Thanks very much.
Thank you.